In this presentation, we will discuss the characteristics of C. botulinum, the mechanism by which the botulinum toxins exerts its effects, and practical uses harnessed by modern medicine. First identified in 1895 by a Belgian microbiologist, C. botulinum gained notoriety due to its role in causing botulism, a serious and potentially fatal illness. C. botulinum is a gram-positive anaerobic endospore-producing bacteria that possesses the characteristic bacillus shape. C. botulinum is categorized within the domain Bacteria, phylum Bacillota, class Clostridia, order Eubacterialis, and family Clostridiaceae. C. botulinum can be further classified based on its ability or inability to lyse proteins and is known to be capable of metabolizing sugar. It thrives in low oxygen environments and grows optimally at a neutral pH and in moderate temperatures. Other notable members of the Clostridia family include C. tetani, the causative agent of tetanus, and C. difficile, known for causing severe intestinal pathology. C. botulinum can affect other organisms within its environment, including humans, through the disease processes that follow from infection. In natural ecosystems, the ability of C. botulinum to decompose proteins plays a role in nutrient cycling. When its spores are exposed to favorable conditions, they will be restored to a vegetative form and will ultimately proliferate and produce those toxins during the sporulation process. The most intriguing aspect of C. botulinum is its production of one of the most potent toxins known to science. Capable of causing the devastating paralytic illness known as botulism, the remarkable potency of botulinum toxin means that it can cause disease in extremely small quantities. C. botulinum produces its signature toxin in the same way that other organisms generate proteins. The central dogma. The protein is coded for in the genetic information of the bacteria, and the transcription process generates messenger RNA that instructs the ribosomes to translate the mRNA into expressed protein. The botulinum toxin acts within the muscular junction to interrupt the normal signaling pathways that produce muscular contraction. But to understand how this happens, we must first understand the neuromuscular junction. The neuromuscular junction is composed of a presynaptic terminal of a nerve axon separated from the motor end plate of a muscle cell by the synapse itself. In order to induce muscular function, the presynaptic neuron must assemble vesicles filled with the neurotransmitter acetylcholine to be transported to the presynaptic terminal for eventual release into the synapse. Once an action potential arrives at the presynaptic terminal, the vesicles fuse with the presynaptic membrane and release their acetylcholine into the synapse. The acetylcholine binds with gated ion channel receptors on the motor end plate of the muscle cell and stimulates a response. The fusion of the vesicle to the membrane involves a protein complex called SNARE, which is generally composed of 3 proteins. 2 of these proteins reside on the interior surface of the presynaptic terminal, while another is expressed on the exterior surface of the vesicle. Under normal circumstances, when this protein complex interacts, a conformational change allows the 2 membranes to fuse and spill the acetylcholine into the synapse. But when botulinum toxin is present, it interferes with this process of vesicle release by cleaving the SNARE proteins, thereby inhibiting their function. When no neurotransmitter is able to be released into the neuromuscular junction, the ability to signal muscular function through ion diffusion is lost, and flaccid paralysis results. Regardless, despite the deadly nature of the botulinum toxin, the medical industry has made great use of it. In 1980, a San Francisco-based ophthalmologist pioneered the use of botulinum toxin for use in a condition known as strabismus, or lazy eye. Botulinum toxin has also become an important treatment option for various spastic disorders, which are characterized by involuntary muscular contractions and stiffness. But by injecting botulinum toxin directly into the affected muscles, spasticity can be reduced. Botulinum toxin is also being employed to manage the pain associated with chronic migraines by injecting small doses of the toxin into specific areas around the head and neck. Of course, while these uses of botulinum toxin are incredibly useful, none of them is particularly well known among the general population. However, there is one use of botulinum toxin that is familiar to most people and which I would consider to be a household name. Botox is a formulation of botulinum toxin which has revolutionized the field of cosmetic dermatology since its introduction and has been used to effectively smooth wrinkles by relaxing the muscles underneath. In conclusion, the humble Clostridium botulinum, a microorganism notorious for its production of highly potent neurotoxin, plays a dual role as a dangerous pathogen and a valuable medical tool, and has been a source of interest since the beginnings of modern microbiology. Initially identified as a source of foodborne illness, the toxins which can cause such harm are used to provide relief and comfort to many. Thank you.